Scientists have identified about 1.4 million locations where single base DNA differences, SNPs, single nucleotide polymorphism. See, polymorphism is a variation at genetic level, pronounced as NIPS. So 1.4 million locations, NIPS, single nucleotide polymorphism. Less than 2% of the genome codes for proteins. The average gene consists of 3000 bases, but sizes vary greatly, with the largest human gene being dystrophin at 2.4 million bases. DNA fingerprinting. DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called as repetitive DNA. Because in these sequences a small stretch of DNA is repeated many times. So identifying differences in these regions is the DNA fingerprinting. Now, there is a satellite DNA. This sequence show high degree of polymorphism and form the basis of DNA fingerprinting. This satellite DNA. As the polymorphism are inherited from parents to children, DNA fingerprinting is the basis of paternity testing in case of disputes. As polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as DNA fingerprinting. Polymorphism is a variation at genetic level arises due to mutations. Since allelic sequence variation also a DNA polymorphism kind of variation. The technique of DNA fingerprinting was initially developed by Alex Jeffrey. He used a satellite DNA as a probe. Now, satellite DNA, it is a DNA polymorphism having high degree of polymorphism. It's a satellite DNA as probe. He used as a satellite DNA as probe that shows very high degree of polymorphism kind of variation.